Hi everyone, good evening guys. Welcome back to Unacademy Meeting channel. Am I visible, audible, all okay? Please let me know. There was a tech issue in the beginning. So yeah, I was trying to resolve it. Good evening all of you. Please let me know audio video normal. Ma'am is back but audio video normal. Yes, it is. Cool. Good evening all of you. Quickly share the session with all of you, all your friends. Quickly do that because guys, this is the first session of last batch for NEET 2027, that is Phoenix 3.0. We have already launched it yesterday, but my first class is today. So yes, as you can see, this is the last batch for NEET 2027, and anybody who is interested and wants to prepare for NEET 2027 in a proper way, focused way with me, complete biology I'll be taking and my Avengers team. So please don't forget to use this offer. This offer is ending tonight. Okay, and now of course you have seen that enough. I will give you more information about this batch at the end of the, this is my team, and we are going to talk more about that later on. All right, yes guys, good evening. All ready? Are we all ready? Quickly give me a thumbs up that you're all set for the first session, and we can start with it. And my first chapter in Phoenix 3.0 batch is molecular basis of inheritance, one of the most important chapters of NEET biology. And from this particular unit guys, we get around 10 to 11 questions every year. This is one of the most highly weighted chapters, molecular basis of inheritance. Yes guys, I want lots of green hearts in the beginning itself. Yes, first of all, I have been able to resolve the tech issue, which I was not honestly expecting, but yes, we have done it. Good evening all of you. All set? All set. Good evening. All right guys, yes, so we are going to start with this chapter, molecular basis of inheritance. Those who have studied the previous chapter, principles of inheritance, with me, fine. Those who haven't, honestly I personally feel that this one chapter gives you a better insight into the previous chapter of principles of inheritance and variation, because that chapter is more like, flashback when we go into the Mendel's era and try to understand what Mendel thought. But it's easy if we understand what we know presently. This chapter will give you the true understanding of the Mendelian era, also Mendelian thought process, Mendelian concepts, right? So one of my favorite chapters. How many of you are excited? How many of you are excited for this? And we can quickly start with that. Let's pray God once that this is our new beginning for NEET 2027 and we are going to rock it together. Okay, this is the first class. The next class will be on the Learners app, Unacademy Learners app, and how all of you can access that, I'm going to tell you that as well. And also a very important piece of information, guys, you have to join me tonight at 12 a.m. and I'm going to reveal that big surprise for all of you. And first, now let's start with this chapter, molecular basis of inheritance. Now we know about, we have heard the word chromosome, right? We have heard the word genes. Now we are going to talk about molecular basis. By the way, what is the physical basis of inheritance? How many of you know that? What is the physical basis of inheritance? How many of you know that? Please let me know in the chat. Physical basis of inheritance is a chromosome. Physical basis is a chromosome. And now molecular basis, or if we say the chemical basis of inheritance, this is DNA. So this chapter is going to be about DNA, which is responsible for inheritance, which is responsible for variations, which is responsible for carrying the characters from one generation to another. Why do we look like our parents and yet different, right? So how do we carry forward those traits over millions or over so many generations? Everything lies, so the answer lies to all those questions in this one chemical, DNA. And DNA is one of the biomolecules, and you must know this is the largest biomolecule. This is the largest biomolecule because this molecule is present in the living beings. So apart from proteins and fats and carbohydrates and so many other molecules that we have within our body, DNA is the largest biomolecule, and it is the master molecule which is going to control every other molecule as well. Which protein will be synthesized, where it has to be targeted, where particular fat synthesis has to take place, everything is going to be regulated by your DNA, right? So this is the master molecule. Everybody, are we all on the same page on YouTube only and in the app? So guys, this is Phoenix 3.0 batch. So the classes will be on Unacademy Learners app. For that, you have to be the subscriber. You have to take a plus batch or iconic batch or the iconic pro batch. I'll be telling you more about it. But first, please understand the chapter, because see, you need to know what you're going to get in those batches. So that is why we are conducting the first live class here. Okay. Now, yes. So starting with molecular basis of inheritance, and I'm going to talk more about DNA. So what is DNA all about? Let's understand. So as you know, I don't like drawing boring notes. So I am going to write all about DNA in this flowchart. Now listen. What does DNA stand for? DNA stands for deoxyribonucleic acid. Deoxyribonucleic acid, and that's how it gets the name DNA, right? Why do we call it deoxyribo? So this gives us an idea, nucleic acid. So first thing, from the name itself, we get to know that it is an acidic molecule. See, that's the beauty of science. The names have deep meaning, right? So from this name deoxyribonucleic acid, we understand this molecule is acidic in nature. It is an acidic molecule. So the acidic nature is because of phosphoric acid, which is a part of DNA. How these components are attached, we are going to discuss it all, right? Second thing, see earlier, and it is called a nucleic acid because it is mainly found in nucleus. It is found in nucleus, therefore it is called a nucleic acid. In fact, I'll tell you, earlier it was known as nuclein because of this reason. Earlier known as nuclein, and this name was given by Friedrich Mischer. This name was given by Friedrich Mischer. Friedrich Mischer called it nuclein because it was found within the nucleus. Is this point clear? Now see guys, in particularly genetics, each and every scientist is super important, and his or her discovery. So please take special notice of this. Is that clear? Now, so later on, it was known as because we found out the acidic nature, it got its name nucleic acid. It got its name nucleic acid. Is that clear? Is that clear to all? So now, deoxyribose. Why do we call it deoxyribose? Let me tell you about the three components which together make DNA. So DNA is further made up of, as I told you, phosphoric acid, which is going to provide the acidic nature to it. This is because of phosphoric acid. Second, why do we call it deoxyribo? Because it has a sugar by the name of deoxyribose. This is the sugar, and it contains 5 carbon atoms. So C5 sugar. So 5 carbon sugar, we can also call it pentose sugar. Is that clear? Is that clear to all? So this is the component number 2. And the component number 3 here is, so it has sugar, it has phosphoric acid, and it has a heterocyclic compound known as nitrogenous bases. These heterocyclic compounds are known as nitrogenous bases. Is that clear to all? Please let me know these points are clear to everyone. I am going to start from 0 and going to take it at the hero level, as I always say. So I know that you are not at 0 level. if you're preparing for NEET 2027 for the first time, you are in class 12 or you are a dropper, but I am going to start from the very basic level for all. Okay, now so there are three components. Is that clear? And we know that these nitrogenous bases, if we are talking about DNA, these are of four types. These are of four types. Or if I classify them broadly into two categories, they are purines and they are pyrimidines. They are pyrimidines. So purines are further of two types. One is adenine, and second is guanine. Right? And pyrimidines, there are otherwise three types of pyrimidine. You can remember it as CUT, C-U-T. But in DNA we are talking about, there are two types of pyrimidines. One is cytosine and the second one is thymine. The third type of pyrimidine is uracil. Now that uracil is found only in RNA. Thymine is found only in DNA. While adenine, guanine and cytosine, they are common nitrogenous bases in DNA and RNA. Please let me know that these points are clear to all. Please tell me how many lectures for this chapter? Around 12 hours for this chapter. We need at least 12 hours to understand it in proper manner. Is that clear? Is this clear? Is that clear to all? Right? Now, understand that these three components, that is phosphoric acid, this sugar and heterocyclic compound, all of them guys are going to combine, in a specific manner. These three are going to combine in a specific manner to form, they combine to form a unit known as, they form one unit known as nucleotide. So the way they are all joined together, this component is known as a nucleotide. Let's see how they are joined together. So I'm going to take it to the next slide and let's understand why we are studying DNA. See, we are studying DNA because it is a molecular basis of inheritance. So it is going to act as a genetic material. We have two types of genetic materials, guys. One is DNA and second is RNA. And if we go by the evolution, this was the second genetic material to get evolved. The first one was actually RNA. Okay, but this was the second one, but the more stable one. See, we have moved towards stability. Is that clear? So it acts as a genetic material. Now, how are these three components joined together? Let's see that. So first of all, we have a sugar and it is a pentose sugar. Now see very carefully. This is sugar molecule and sugar is pentose sugar. What do we understand by pentose sugar? There are 5 carbon atoms. 1, 2, 3, 4. And what about the fifth carbon? The fifth carbon goes here. This is the fifth carbon, CH2OH. This is the fifth carbon. This is your pentose sugar. Now, here remember, every carbon atom has a significance. Towards the carbon atom number 1 is attached nitrogenous base. So with carbon number 1 is attached nitrogenous base, and with carbon number 5 is attached the phosphoric acid. I will symbolize it with P encircled. Is that clear? Now, see the bonds that have been produced. The bond between sugar and nitrogenous base, it could be any A, T, G or C, right? Now, nitrogenous base and sugar, this bond is known as the glycosidic bond. So there are numericals also around this topic. Calculate the number of bonds. So you should know glycosidic bond. Then the bond between carbon number 5 and phosphoric acid, this is known as phosphoester bond. Phosphoester bond. Please tell me these points are clear, right? Now, see, actually, as I said, every carbon has a carbon here in the sugar has a very important significance, very important value. The value of carbon number 1 is that it is attaching nitrogenous base with itself and forming the glycosidic bond. What about carbon number 2? This carbon number 2 actually decides whether it is a ribose sugar or it is a deoxyribose sugar. See, the only difference between, see, actually it is HOH, right? So The difference between ribose sugar and deoxyribose sugar is only at carbon number 2. If it is an RNA sugar, that is ribose sugar, it would be OH here. HOH. That would make it as a ribose sugar, the only difference. But if I remove O, deoxy, that means if I remove one oxygen, it becomes a deoxyribose sugar. So the value of this carbon number 2 is, it determines whether it is going to be a deoxyribose sugar or a ribose sugar. What is the significance of carbon number 3? Just wait for it. Carbon number 4 is already holding on to carbon number 5, and carbon number 5 is holding on to phosphoric acid, forming the phosphoester bond, right? Now this one unit, look carefully, this one unit is known as, a nucleotide. This is a nucleotide. Is that clear? This is a nucleotide. So guys, nucleotide is made up of 3 components. It has phosphoric acid, then sugar, and then nitrogenous base. Is that clear? So, and now nucleotide is called the unit of DNA. It is a unit, or you can say the monomer of DNA. Is that clear? To be precise, you can call it deoxyribonucleotides because it is a nucleotide of DNA. It can also be known as, aka known as deoxyribonucleotide. So that means if you keep on joining these nucleotides one after the another, right, this is going to form a chain of DNA. Let's do that. So if I attach one more here, let's say, and now I'm going to ask you a numerical right after this, so please pay attention. This is nitrogenous base, another nitrogenous base, and this is CH2OH. This is carbon number 5, and here we have phosphoric acid attached. How many of you are clear with this? Is that clear? Now this is another nucleotide. Now one after the other, such nucleotides will keep on getting attached, and it is going to form one DNA chain. Now how are they going to get attached? Now please notice that. See here. So this carbon number 3 of the previous nucleotide is getting attached to the phosphoric acid of next nucleotide. Do you understand this? And now this bond formed between the 2 nucleotides, guys, this is known as phosphodiester bond. Are the bonds clear to everyone? Please let me know. Are these bonds clear to all? Please tell me, guys, are these bonds clear to you? Now, because there are 2 nucleotides attached here, can I say this is a dinucleotide? Then, if there are 3, it will form trinucleotide. Yes or no? And similarly, if there are so many, can we call it a polynucleotide? So guys, understand that DNA basically is nothing. So DNA is nothing but a chain of so many nucleotides attached together. Can we say that? Please let me know. So DNA is what? DNA is basically a chain of, it is a chain of, poly means many nucleotides held together. And what is the bond between many nucleotides? Which bond connects various nucleotides? Just have a look. It is the phosphodiester bond. Is this point clear? Now, if I have a question for all of you. Since there are 2, so I can call it a dinucleotide. This is a dinucleotide, means 2 nucleotides attached. Now, if I ask you, calculate the number of, yes, now guys, I want your answers here. Calculate, this is a question, calculate the number of, number 1, glycosidic bonds here. Quickly give me an answer here. How many glycosidic bonds do you see in this structure? Let me know the answer here. And then number of phosphoester bonds. Yes. Number of phosphoester bonds. You have to answer in the chat quickly. So Shivani and Kiminson, Ruchir, Pramod, everybody is able to say, you are saying that glycosidic bonds are 2 in number. There is one between this nitrogenous base and sugar, and second is here. So glycosidic bonds are 2 in number. Very good, very good. Now, if you can answer this question, then it means you are actually understanding the topic. Phosphoester bonds. Yes, guys. How many phosphoester bonds do you see here? How many phosphoester bonds do you see here, guys? How many phosphoester bonds do you see here? You are saying 2. You are saying 2. What if I say answer is 4? What if I say answer is 4? Yes, please. Answer is 4. Let's understand. This is phosphodiester bond. One phosphodiester bond is equivalent to, this is equivalent to 2 phosphoester bonds. So 2 phosphoester bonds here, 3 and 4. Please tell me if you are clear with this point. Please tell me if you are clear with this point. Is that clear to all? Is that clear to everyone? That's the point here. Is that clear? So if you are asked only about phosphodiester bond, answer would be 1. If you are asked to calculate the number of phosphoester bonds, you have to include this phosphodiester bond, and 1 phosphodiester bond is equivalent to 2 phosphoester bonds. Please tell me that this point is clear to everyone. Yes, please. Clear to all? Noted? Right? Okay. Now, let's talk more about DNA. By the way, guys, this DNA can be, it can be single-stranded. That means just single thread can act as a genetic material in some organisms. So we call it ssDNA. While in most of the organisms, it is double-stranded. It is double-stranded. That is dsDNA. Is that clear? Now, which organisms have single-stranded DNA? Which one have double-stranded? We will come to the name. We'll come to that topic also, and remember the names as well. We'll come to that gradually. So, guys, this is a dinucleotide that I have drawn. Now, similarly, if I keep on, see, I'm going to draw few nucleotides. So, this is sugar. Let's make it a little big. This is the sugar, right? I'm going to draw one more. This is sugar, one more, and sugar, right? This is CH2OH, the fifth one, CH2OH, the fifth one, and so on here, right? Let's join some other components. At carbon 1, we have nitrogenous base, nitrogenous base, nitrogenous base, right? While at carbon number 5, phosphoric acid is attached, and they are all attached to one another through this phosphodiester bond. Is that clear? Now listen, if we talk about the double-stranded DNA, this is one strand. Strand means one thread of DNA. Similarly, we have another thread of DNA right opposite to it. But now I'm going to come to the structure. First, see. Now, the strand which is present right in front of it it is parallel, but the polarity is opposite. So we say the two strands are anti-parallel. And see, the anti-parallel nature is because of this sugar. Here, see the polarity of sugar. 1, 2, 3, 4, and 5. Right? Now, the sugar in the opposite strand is going to be opposite. The polarity will be just opposite. See here. Now, oxygen is on the upper side. Rather, it's going to be like this. So, the polarity of sugar will get reversed. So, this is carbon 1, 2, 3, 4, and 5. Do we understand this? Similarly here. So, it's going to be like this here. So do we see that in the opposite strand of DNA, the polarity of sugar has been reversed. So this is carbon 1. This is carbon 1, and here the nitrogenous base will be attached. Is that clear? And now talking about phosphoric acid, that will be attached to the fifth carbon only, like this, and they will all be attached through carbon number 3, Please tell me these points are clear to everyone. Noted? Okay. So the two strands are going to get connected through, they will be connected through this nitrogenous base. The two DNA strands are connected through nitrogenous base, and the bonds formed between them are called hydrogen bonds. They are called hydrogen bonds. How many hydrogen bonds? Now that depends upon the nitrogenous base which is present here. See, remember, adenine will always bind to thymine, and guanine will always bind to cytosine. It will always bind to cytosine. Is that clear? Now, calculating the number of hydrogen bonds. See, as a fact, adenine will always bind to thymine through 2 hydrogen bonds, and guanine to cytosine through 3 hydrogen bonds. So 2 hydrogen bonds here and 3 hydrogen bonds here. Is that clear? Again, we have a numerical. You can be given that there, in a certain DNA strand, there are so many base pairs. So see, these two together make one base pair. The two nitrogenous bases together form, this is one base pair. So if you have been given the number of base pairs, let's say there are 200 base pairs. Base pairs are written as BP. Okay, so 200 BP, that is base pairs, out of which 40 are adenine. 40 are adenine. Can you calculate the number of cytosine? See, again, let's use basic maths here. So we know that if these are 40 in number, and these will also be 40 because adenine will always bind to thymine, yes or no? So 40 adenine means 40 thymine. So total it would be 80. Remaining, if the total are 200, what would be the remaining? Anti-parallel in nature. What is the reason behind anti-parallel? Is because of opposite polarity of sugar. Please tell me these points are clear. Then third, we have seen that DNA is formed of the monomeric units, the monomers. The monomeric unit of DNA is, yes guys, monomeric unit of DNA is deoxyribonucleotides. Please tell me all these points are clear to everyone. Yes, please. Is that clear? Is that clear to all? Right? Now, understand that in this structure, sugar and phosphate, these two structures, they are going to form the main backbone of DNA. Sugar, phosphate, right? So, sugar and phosphate form the backbone. Write down. Sugar and phosphate, these two structures, they form the backbone of DNA. They form backbone of DNA. Yes, please. Are these points clear? Sugar and phosphate form the backbone of DNA. Yes, guys. Please tell me these points are clear to everyone. Can't we say DNA is parallel in nature? Riya, of course they are parallel. And why are they? See, there are two things. They are parallel and they are anti also. The anti word comes because of opposite polarity. And it is of course parallel. Now, let's understand why they are parallel. See, why they are parallel. For that, you must know the structure of purine and see. we say the two DNA strands, they are connected through nitrogenous bases. Now have a look. I was coming down to this point. A will always bind to T, G will always bind to C. If you see the nature, this is also a purine, while this one is a pyrimidine. This is purine, this is a pyrimidine, right? Now, if we know the structure of purine and pyrimidine, let's see nitrogenous bases first. So, we have purine, two types of nitrogenous bases, purines and pyrimidines, right? Understand the structure here. See, pyrimidine, which can be CUT in general. So, pyrimidine is a 6-membered structure. It is 6-membered and there is single ring. So, if I draw a skeletal structure, it is like this, right? This is a basic ring, 6-membered ring. Purine, while there are two purines, A and G, adenine and guanine, the purine is a 9-membered structure and there are double rings. There are two rings, double ring structure. So it's like this. Okay, it's like this. 1, 2, 3, 4, 5, 6, 7, 8, 9. There are 9. Now, I am saying, as you can see in the structure, purine will bind with pyrimidine. So, this is a bigger structure, this is a smaller structure. Bigger structure, smaller structure. Or it can reverse. This side you have pyrimidine, opposite side you have purine. Point is, purine will always bind with pyrimidine. So, one is bigger, another is smaller. See the adjustment of the structure here? So, that provides parallel nature. What is the reason behind parallel nature? Parallel nature is because purine always binds to pyrimidine. Please tell me this point is clear to you. So one side we have a bigger molecule, on the opposite side we have a yes, that is anti. Anti is because of opposite polarity of sugar. Anti means the 3 dash end, in front of 3 dash end we have 5 dash end, and on the opposite to 5 dash end we have a 3 dash end. Is that clear? So that's what anti-parallel nature is. But it is anti because of opposite polarity of sugar. Sugar is responsible for the anti nature. But parallel, the width remains the same. They will never converge because purine will bind to pyrimidine. Please tell me these points are clear. Yes, guys. Is that clear to everyone? Clear to everyone? Now, you should know that this whole information, the structure of DNA, it was decoded by, understand, this was explained by or deciphered by X-ray diffraction. We got all this information because of X-ray diffraction method. X-ray diffraction. This was done by Morris Wilkins and Rosalind Franklin. Wilkins and Franklin. Is that clear? So these are the two people who did this X-ray diffraction. Okay, and this X-ray diffraction was decoded by, deciphered by, read by Watson and Crick. So that is why Watson and Crick are given this credit to understand or decode the structure of DNA. These are the two people, Watson and Crick, because they basically deciphered this X-ray diffraction. So the technique used is X-ray diffraction. Who did that technique? Who were the physicists who did that X-ray? These two were the physicists, Wilkins and Franklin. But that X-ray was read by Watson and Crick. See, if there is an X-ray in front of you, you and me, we won't be able to decipher that, what is being shown. We might see a general crack, but what type of it is, we can't do that. But people who can read it, they were Watson and Crick. Please tell me these points are clear to all. Is that clear? Is that clear? Right? All right. So Watson and Crick Watson and Crick in 1953, they decoded the structure of DNA. Please tell me these points are clear to all. Please tell me these points are clear to all. Now, according to this, guys, according to this, DNA, according to these people, DNA, see, DNA has a double-stranded. DNA is double-stranded. I've already informed you. DNA is double-stranded. This is the first information that they gave. Anti-parallel. The monomeric units are this. Sugar and phosphate forms the backbone, right? And next point, DNA is helical. DNA is helical in nature. Helical in nature. What do we understand by helical? Helical nature. So these two strands are not actually straight lines like these. They are not in straight manner. So the helical is like a, rounded staircase. There are two threads which are running parallel to each other. Are you getting that? So actually, let's say this is how DNA. These are the two strands. Just ignore the circular ends. So these are the two strands and they are running helical. Are you getting it? Like a jalebi, right? So that's how the structure of DNA looks like. So if I draw the helical nature, I'll draw it on the next slide. See, this is one thread, one. This is helical. This is one thread. I'll label it as 3' end and 5' end. And let me draw another. This is going parallel like this. This is 3' end, this would be 5' end. These are the two strands. Are you getting it? So imagine they are wrapping, and yeah, let's say this is a common axis, and around this common axis it is getting wound. So this is called the helical nature. And as a matter of fact, we have different forms of DNA. We have overall 5 different forms, but the most common form of DNA, most common form of DNA is bDNA. This is bDNA. Okay, and bDNA is a right-handed helix. It is a right-handed helix. See, goes like this, clockwise direction. Is that clear? Now, this is called the minor groove. Groove means a depth, right? And then we have this major groove. We have this, wait. We have this as a major groove. Major means bigger groove. Okay. Another point. See. So whenever we are, now understand this point, like twisted. Yes, exactly twisted. So I am sure we see when we try to wind something, let's use a prop here. Yeah, we have a prop here, an unused cable. So let's say I have to wind this wire on around my wrist. So I started from here, right? I started from here and I go back. So the place where it is completing one circle, okay, let's start it from the end. So the place where it is completing one circle, the complete one revolution, right? This whole length is known as, this whole length around my wrist. So one complete revolution is known as pitch. Okay. Similarly, so it starts from here and goes till here. That's where one complete pitch is, one complete revolution. This is known as pitch. This is known as pitch. Are we clear? So listen now. So for bDNA, I am giving you all the information regarding bDNA. All the data I am going to write is true for bDNA. So in this one complete revolution, this is known as pitch. Is known as pitch, and pitch is equal to 3.4 nanometer. What is the length of the pitch? It is 3.4 nanometer. Another point. In this pitch, there are 10 base pairs. There are 10 base pairs, I am saying. So read it carefully. 10 base pairs means, let's say we have A here and T here, G, C, G, C. It could be anything. That depends. A, T. So fifth one, sixth, seventh, eighth, ninth, and 10th. Do you see? I have not drawn in a very regular manner, but the point is, in this one complete pitch, there will be how many base pairs? 10 base pairs. How many bases? 20, of course, right? So see, 10 base pairs means, 10 base pairs here means this one. Sorry. Yeah. So this one is one base pair. Are you getting it? This is another base pair, third base pair, and so on. So one base pair, as you know, has two bases. Please tell me these points are clear to all. Please tell me this point is clear to all. Is that clear? Noted? All right. So what is the value? 3.4 nanometer. Now just use simple mathematics and let me know. Use simple mathematics and let me know if the total length from here to here is how much? 3.4 nanometer, and there are 10 base pairs. What is the distance between two bases? What is the distance between any two bases? Hashtag distance between any two bases is known as rise per base pair. Rise per base pair. Yes, tell me. What is the distance between any two bases, guys? This divided by 10, will it be 0.34 nanometer? Yes or no? The distance between any two bases would be 0.34 nanometer. Yes or no? Now we are going to use this information. Understand, with this information, you can calculate the length of any DNA. If I tell you there is certain number of base pairs present in a DNA, can you calculate the length of the DNA or not? Can you calculate the length of the DNA or not? Yes, guys. Quickly. For example, from this information, I have been able to calculate the total length of human DNA if the number of total base pairs, see, if the number of base pairs in diploid content, 2n content is equal to, yes please, 6.6 into 10 raised to power 9 base pairs. If I tell you the 2n content of DNA has these many base pairs, can you calculate the total length of DNA? Let me know, guys, quickly. This is another numerical. So we are going to solve numericals side by side. This chapter is full of numericals. Lots of concepts, lots of numericals here. Yes, guys. Ashrita says 2.2 meters. What have you done? Very good, Ria. You're perfect, perfect. So how do we calculate? So if the distance between two base pairs is how much? 0.34 nanometer, and there are total number of, and the total number of 2n content is 6.6. All you have to do is 6.6 into 10 raised to power 9 into 0.34 nanometer. So I'll convert this into meter by multiplying it with -9. Yes, guys, that's how it is. So -9 and this will get cancelled. Can you calculate? It will come out to be 2.2 meters. So the length of total DNA in your each cell, so the entire 2n content, all the 46 chromosomes, the total DNA of 46 chromosomes, when joined together, will form 2.2 meter of length. And similarly, if I give you just haploid content will be half of it, 3.3 into 10 raised to power 9. What will be the length? Length would be how much? Half of this. 1.1 meter? Please tell me that you are clear with the concept. And similarly, can you calculate the length of any DNA just by using this information? And all this information was decoded by these two people by studying the X-ray diffraction method. And understand that the structure of DNA which was given by Watson and Crick was a two-dimensional structure. It was a secondary structure. They gave secondary structure of DNA. Secondary structure of DNA means two-dimensional structure. Please tell me these points are clear to all. Please tell me these points are clear to all. Right? Anyone with any doubt until here? Yes, everyone. Please tell me these points are clear to everyone. Noted? Now, I want to tell you one more thing. That in order to understand the structure of DNA, Watson and Crick, of course, majorly depended on the X-ray diffraction. That X-ray, okay, which was done by Wilkins, Morris Wilkins and Rosalind Franklin, two people, okay. But they also used some other information. What was that information? They used the information given by another scientist, Chargaff. By Chargaff. So, Chargaff has already given certain information about DNA and we call it Chargaff's rules. What did Chargaff said? Chargaff has already announced these things. What did Chargaff said? Listen, Chargaff said that because purine always binds with pyrimidine, right? Therefore, the first thing, amount of purine in DNA will always be equal to amount of pyrimidine. See, of course, we are talking about double-stranded DNA, right? So, amount of purine will always be equal to pyrimidine. We also know that adenine binds with thymine. So, can we say amount of thymine is always equal to amount of adenine? This was all already known to Chargaff, and Watson and Crick had used his information, right? Then third, amount of guanine will always be equal to amount of cytosine. We have already solved one numerical also using Chargaff's rules. So Watson and Crick has used multiple sources of information to give us that structure. Is that clear? Now, the total amount of purine, see, amount of purine is equal to amount of pyrimidine, T is equal to A, G is equal to C. So, can we say, if amount of purine is equal to pyrimidine, can we say, now see, that A plus, sorry, can we say purine, sorry, I'll write it this way so that we can, yeah, now it's fine. So can we say purine, the amount of purine, so A plus G is equal to T plus C? Tell me if you are able to understand this. Now I'm going to write down two ratios. I'm going to write down two ratios here. I'm going to write down two ratios here and you have to take clear notice of it. Please, please, please. I need your focus here. Now, first of all, A plus G, that is total purine will be equal to total pyrimidine. Can I write this first equation like this? And now, can I write this equation as A plus G upon T plus C is equal to 1? That is simple maths. So, whenever you bring it on the other side of the sign of equality, it goes in the denominator. Is that clear to all? So this is purine upon pyrimidine. Can I say purine upon pyrimidine is equal to 1? Please tell me. Are you able to understand all these points? Everyone, please tell me. Is this clear to all? Right? Right? Yeah. Now, very, very important, where you are going to make a mistake, every chance of you making mistakes. So, I am going to say that A plus T Now see what I'm going to write. A plus T upon G plus C is constant for a species. Is constant for a species. I want everyone to stop writing for a while. Look at this equation, think over it, and see if you are able to see the difference between the two. Of course, I'm right here to explain, but are you able to see the difference? What am I writing? A plus T upon G plus C. I'm saying A plus G. Here I'm saying A plus T. What does this mean? This means, guys, this means that let's say this is one DNA strand, this is another DNA strand. This is a DNA of a particular species. Now, how much AT content will be present and how much GC content will be present, that is fixed for every species. That is fixed for every species. How much AT is present and how much GC, how many such GC bonds and how many AT bonds will be present, that is fixed for a species. So all of us has a fixed percentage of AT and GC content, and our value will not match with any other species. Please tell me these points are clear to everyone. Anyone with any doubt? Is it true that AT is more than GC? We'll see that. We'll see that. We have to learn so many things. We have to learn so many things. Yes, guys. Are these points clear? See what is true and what isn't true. We'll keep on learning. These are certain things which Chargaff has announced. Till date, we have made so many advancements, right? Human Genome Project has already been successful, and Human Genome Project itself has given us so much information about our genome. And still, new discoveries are getting made every day. While we are busy preparing only for a NEET exam, there are new discoveries happening every day. So gradually in this chapter, I'll keep on decoding. I don't want to announce any other information because I am just talking about Chargaff. This is what Chargaff said. Please see that. This is what Chargaff said. But otherwise, yes, you are right. In eukaryotic DNA, generally, AT content is more. But again, how much will be present, that is different for every species, and the amount is fixed. That is fixed. Is that clear? Is that clear? So this is Chargaff's rules, and we can solve so many numericals on this topic. Please tell me these points are super duper clear. Anyone with any doubt? Anyone with any doubt? Right? Now, there is one more information that you should know. See, regarding nitrogenous base. So we know that nitrogenous base will bind with deoxyribose sugar or any pentose sugar. It could be ribose also. And these two components will together form sugar, and this will form nucleoside. While if I add phosphoric acid to this, it will form nucleotide. Please tell me these points are clear to everyone. Yes, please. Are these points clear? Are these points clear? And I'm going to write nitrogenous bases here. Now see, guys. Everyone, please pay attention. We have different names for these nucleosides and nucleotides. See, first of all, if you bind nitrogenous base with sugar only, it will form nucleoside. SS together. Bind sugar to nitrogenous base, it will become side. And if you add the third component also, when there are three components bound, 3 and T. Can you remember these terms? Very basic. It might look easy, but when you are sitting for an exam, even the simple things go wrong. And remember, you are going for not a normal exam in NEET 2027. This NEET 2027 is about CBT, and CBT is not going to be as easy as you think. And by the way, guys, what you are watching is Phoenix 3.0 first batch. The batch has to be taken. The first class is free, and tonight at 12 a.m. you have to join me to understand how you can watch another 3 lectures also absolutely free. For that you have to join me at 12 a.m. but otherwise the batch is for 4,499, but this offer is going to end tonight. So if you want to buy it at this price, you have to use this code and buy it today itself. Now listen, if you have already taken the exam in 2026, if you're a dropper and you have scored good, so this is the scholarship available, and all these are available only for tonight, till tonight. Okay, so if your score is below 450, you can avail a scholarship of 60%, and similarly as your score increases, if you're scoring above 550, you can avail scholarship up to 90%. You have to share your scorecard along to avail the scholarship. So this is a great offer. So this is the wall of fame, all these students, and we have, see, 4499, this is plus. But if you want more facilities like one-on-one mentorship, you want NEET UG Assure, and you want printed notes, you have to go for Iconic, right? And if you want CBT practice as well included in that, then we have Iconic Pro, and you will be joining the 100 club. Is that clear? So the subscription plans are different accordingly, and yes, if you are already a subscriber, I'm sure many of you have already subscribed. You are watching this session with me. Many of you have already subscribed. So if you want to upgrade your plan for CBT preparation, this is the price. So online proctor test, if you want online CBT, it is for 2,999, and if you want offline, personally I feel you should go drop down to the center and get a total feel, 5,999. Okay, so join that. And this is very important. The 2nd August CBT is absolutely free. Those of you who still cannot understand the importance of CBT and don't understand the difference, how the preparation would differ and how the examination will differ, how your performance will differ so much from a typical pen and paper mode and the CBT mode, take this absolutely free CBT on 2nd August. There's a link given in the description box. Click on that, register for it and see it for yourself. You will see absolute difference. Your performance is going to decrease so much in the beginning. You might feel disheartened also. What has happened? I knew the chapter, still such low score. So you see it for yourself. The only thing is, this is all because of the change in the interface. So every preparation requires a different methodology. Now coming back to the topic here. So we have nitrogenous bases. Number 1, we have adenine. This is a purine. We have guanine, again a purine. Then we have cytosine. Then thymine, and I'm going to tell you about uracil also, although uracil is not a part of DNA. This is only in RNA, while thymine is only in DNA. Is that clear? While rest of them, they are common to DNA and RNA. So the sugar which can attach to DNA can be deoxyribose also, if it is DNA, and if it is RNA, it is a part of RNA, then it will bind to ribose sugar, but it can bind to both. That is why I'm putting a sign of oblique, deoxyribose or ribose, deoxyribose or ribose. While thymine is a part of only DNA, so it will only bind to deoxyribose sugar. And similarly, uracil is only a part of RNA, so it can only bind to ribose sugar, right? Now, if the nitrogenous base is attached to the sugar, it will form a nucleoside, right? And the nucleoside made by the combination of these two will be known as adenosine. Very important. What is the difference between adenine and adenosine? Adenosine is a nucleoside. Adenosine has adenine along with sugar. It is attached to the sugar. Through which bond? Let me know. Which is the bond between these two? Which is the bond between these two? Yes, guys. Which is the bond formed between nitrogenous base and pentose sugar? Quick, quick, quick. Everyone, would you be sending the telegram? Yes, Kimi, I'll be sending these notes on the telegram link. Glycosidic bond. Very good, very good. All of you, all of you. So there's a glycosidic bond formed between adenosine. Similarly, we have guanosine. See the difference. Guanine becomes guanosine by addition of sugar. Then cytidine, right? Then thymidine and uridine. And now if I add phosphoric acid also to all of these, by addition of phosphoric acid, it becomes an acid now. It becomes nucleotide. So it will become adenyllic acid because phosphoric acid is attached, adenyllic acid. Similarly, guanyllic acid, cytidyllic acid. So you should be able to differentiate between these terms. Thymidyllic acid, you can get a question. Which of the following is a correct set of nucleotides or a correct set of nucleosides? You should be able to differentiate between these terms. And uridylic acid. We have another name for these two also. Because we are adding one phosphate to adenosine, can I also call it adenosine monophosphate? Yes or no? Just the same name of nucleoside with monophosphate. Similarly, this can be known as guanosine monophosphate. Yes or no? Cytidine monophosphate, thymidine monophosphate, uridine monophosphate. Please let me know that you have understood this point. Yes, guys, let me know you have understood this point, everyone. Right? Hi, Satyam. Good evening. You've joined quite late. What happened? Right? Cool. So, this was all. And next, coming on to the next aspect. Now, gradually, we are going to get into the deeper aspects of DNA. The next thing is packaging of DNA. Yes. Very important topic. Packaging of DNA. Now listen. We have already understood what is the length of DNA. Let's go back here. Yes. So we have understood that because we are majorly talking about bDNA as it is the most common form of DNA. There are 5 different forms actually. A, B, C, D and Z. Okay. Now, bDNA is the most common right-handed helix and the distance between two base pairs is 0.34 and that's how we have been able to calculate the total length of DNA, right? So, let's write that down again. So, the total length of DNA. I am talking about the 2n component is equal to, how do you calculate? Number of nucleotides into rise per base pair, that is 0.34 nanometer. This is the formula. This is the formula. Use this formula. So, if you have information about the total number of nucleotides, you can calculate the length of any DNA. As far as human DNA is concerned, write down. Let's write down. For human DNA, the 2n is equal to 6.6 into 10 raised to power 9 base pairs. This is the total number of base pairs. And for haploid content, it would of course be half of it, 10 raised to power 9 base pairs. So that means, therefore, length is equal to, we have already calculated, right? Into 0.34, length would be 2.2 meters, and the length of, this is for 2n content, and length for haploid content would be 1.1 meter. This is n content. But because we have 2n content in most of the cells, this is what is important for us. Now listen. So each cell has this long DNA. 2.2, if you want to imagine, 2.2 is almost equal to the size of a dupatta. Do you understand? That is the size of almost a dupatta. And imagine such a long DNA is present in a microscopic cell. Your cell is not visible, any cell generally, like a cell is not visible to you. Your skin cells or any cell is not visible to you. But you have 2.2 meter of DNA in that cell. How is such long DNA fitting into a microscopic structure? And on top of it, if you see the structure of nucleus, the size of nucleus, listen, the size, this fits into a nucleus with the size 10 raised to the minus 6 meter. This is a rough size of 10 raised to the minus 6 meter. 2.2 meter fitting into 10 raised to minus 6 meter. How is that possible? That brings us to the next topic. How is DNA packed within the cell? All right. Now, understand that DNA is a acidic molecule, and neither acid nor base, when they are alone, they won't be stable. So if we want a stable system, then acid should be associated with a base, and similarly base should be associated with the acid. Now we are eukaryotes, right? How is DNA stabilized? So DNA, in order to stabilize DNA, see, DNA is acidic molecule, right? Why is it acidic? It is acidic molecule because of phosphoric acid, right? In order to stabilize, it is associated with, therefore, it is associated with basic proteins. Okay, it is associated with basic proteins. First of all, this is associated with basic proteins, right? Now, as far as prokaryotes are concerned, in prokaryotes, there are some amino proteins. These amino proteins are basic in nature. Okay, and these amino proteins are also in the form of hooks. They occur as hooks. So see, I'll show you. Let's say this is a prokaryotic cell, and we know DNA is randomly placed in the cytoplasm, right? But in the cytoplasm are present these hook-like amino proteins. Okay, and on these hook-like amino proteins, our DNA is wrapped, like prokaryotic DNA is wrapped on it. It is circular, there are no free ends, so wherever it started from, it came back to it, so becomes circular. It is double-stranded, of course, so there are two strands like this. There are two strands, double-stranded, but it is attached to these hook-like amino proteins. These, are you getting it? And these hook-like proteins are able to stabilize this DNA, and it is a very small amount, so this serves our purpose. Is this point clear? Is this clear to all? Yes, Harita, one pitch. Whenever we are talking about one pitch, we are talking about the 2N content only. All right. So these are amino proteins. Is everyone able to understand? Thank you, Maruful. If you are liking the lecture, that means you are also studying it deeply. It's important to be very focused to be able to appreciate a lecture. Thank you. And I hope everybody is able to understand. So if you're able to understand, don't be miser in sending green hearts. Okay, understood? All right, guys, cool. Now, so this is about prokaryotes. Now, if I talk about eukaryotes, in eukaryotes, guys, there are special basic proteins known as histone proteins, histones. Now, histones are again basic proteins, but these are found only in eukaryotes, while amino proteins are found in prokaryotes. Is that clear? And now if I particularly talk about arc bacteria, so there will be more differentiation. Arc bacteria also comes under prokaryotes, the most primitive prokaryotes, right? Bacteria. So their basic proteins to stabilize DNA, they're very similar to histone proteins. They have difference too. So the arc bacteria have basic proteins which are similar to histones, but not histones. Histones are found only in eukaryotes. Okay. Now this is a special category of basic proteins. So these are basic, of course. And why they are basic? See, the nature of protein will ultimately depend upon the nature of amino acid, right? So they are rich in, it is rich in lysine and arginine. These are two basic amino acids. So if there are more basic amino acids, the overall nature of protein will also be basic. Please tell me these points are clear. So it is rich in lysine and arginine. Please tell me these points are clear to everyone, right? So rich in lysine and arginine. Clear to all? Noted. And these histones, they form an octamer. They form an octamer. Octamer means 8 molecules of histones will club together. How? I'm going to show you on the next slide. 8 molecules together. Is that clear? And it is a class of proteins. We have further types of histones also. We'll all discuss that. Please tell me these points are clear. Noted? Now, let's talk about the histone octamer. See, in an octamer, let's see this. So we have this octamer, and in this octamer, guys, there are 4 types of histones, which are H2A, 2 molecules of H2A, 2 molecules of H3, similarly 2 molecules of H2B, then 2 molecules of H3 and 2 molecules of H4. 4 different types of histones will form a histone octamer. This is a histone octamer. Clear? This is a histone octamer. And what all histones are combining together? So H2A, H2B, H3 and H4. These 4 types. How many? 2 molecules of each coming together to form an octamer. Please tell me these points are clear to all. Noted? Similarly, okay, now we'll have another octamer like that, and still another octamer, and so on. And what about DNA? How is DNA placed now? So I'm going to show you DNA here. See, now DNA will be binding on this, like that. Okay, and then like this, and this, and this, and this, and it goes on and on. Do we understand? Please tell me, do you understand this? So what you see here is DNA. This is the DNA. So DNA and histone octamer. In total, what we have formed here, this one is called a nucleosome. What do we call this? Similarly, this is also a nucleosome. This is another nucleosome. Do we understand this? This is a, I'm writing here, this is a nucleosome. So nucleosome is called, this nucleosome, yes, it is called a repeating unit of DNA. This is a repeating unit of DNA. Please tell me that you understand this part. And what is nucleosome made up of? Nucleosome consists of DNA plus histone octamer. DNA plus histone octamer. Please tell me these points are clear to all. Right? Now, one more aspect, particularly this part of DNA, which is combining or which is connecting the two nucleosomes. Listen, this part of DNA which is connecting the two nucleosomes, this is known as linker DNA. This is known as linker DNA. Easy to understand. The part of DNA, now this linker DNA is not a part of nucleosome. Note that. It is not a part of nucleosome. But it is of course a part of DNA, right? So we need to stabilize this region also, right? So here, guys, we have another histone molecule in order to stabilize this. So this is another. So this is DNA. This is all linker DNA, and linker DNA is stabilized by another histone called H1 histone. So H1 histone stabilizes what? It stabilizes linker DNA. Please tell me these points are clear. Please tell me these points are clear. Can you complete the chapter on YouTube? Ratna? Thank you so much, first of all. But for all those of you who are so interested in learning more about this, I am going to give you a great opportunity. For that, I'm going to reveal that opportunity tonight at 12 a.m. How you can complete this chapter with me, like free of cost, you will be able to understand tonight at 12 a.m. So don't forget to join me then. Okay? I know you are very curious to complete this chapter, and I'm going to make it buttery easy for you. Okay? That's my promise. So first tell me, are these points clear to everyone? Does histidine is not a part of it? Ria, of course, histidine may be present, right? You're talking about this histone protein. There are other amino acids also present, but the most occurring amino acids which actually give it the basic nature, that is arginine and lysine. Okay, so guys, so it means H1 histone is not a part of, hashtag not a part of histone octamer. It is not a part of histone octamer. This is going to stabilize linker DNA and not rest of the DNA. Please tell me these points are clear to everyone. Yes, guys, is that clear to all? And actually, this is a double-stranded DNA that I have drawn here. Is that clear? This one thread represents a double-stranded DNA. Please tell me this is all clear. Noted? Yes, guys. Is that clear to all? Anyone with any doubt? Yes, please. All clear? Okay, Krish, I'll do something about this. Thank you, Maruful. Thank you. Now, as a matter of fact, I'll tell you some facts about nucleosome also. This nucleosome has around 200 base pairs of DNA. This is also an important piece of information that you must keep. Each nucleosome, how many base pairs in this one nucleosome? 200 base pairs of DNA. Are these points clear to all? Yes, please. This linker DNA is also double-stranded. Everything is double-stranded. We are only talking about double-stranded, guys. This is all double-stranded DNA. Is that clear? This is all double-stranded DNA. Clear to all? So let me try making double-stranded. So it would be like this, like this. So it is double-stranded. It is all double-stranded. Okay? I didn't want to. It is all double-stranded. Just understand it like this. Even linker DNA is double-stranded. There is nothing single-stranded. Clear to all? Right? Okay, Aftab? All right, guys. All right. So, similarly, so how would overall, see, now listen, how would it look like? So, this is one nucleosome, this is another nucleosome, this is these are histone octamers that I'm drawing, and this is the DNA. That's how it looks like, right? So that's how it looks like, and it is double-stranded like this. Clear to all? It's all double-stranded like this. I hope this is clear now. Anyone with any doubt? And so on. Is this clear? So this structure, so under, now see, this structure, this whole structure is known as chromatin. This whole structure is now known as chromatin. This is called chromatin. So these are all nucleosomes. These are all, this is nucleosomes. Is that clear? So nucleosomes will give it a, they will form chromatin. Let's say this is nucleosome 1, this is nucleosome 2, nucleosome 3, histone plus DNA, right? That is nucleosome 4, nucleosome 5, and so on. Is that clear to everyone? Right? And now this gives it beads on the string appearance. Chromatin has, this is called, it has a beads on the string appearance. Is that clear? Is that clear to all? Now normally our DNA is in the form of a thread. Remember that chapter? Mitosis and meiosis. So you always draw it like this. What does this represent? So basically this represent, these are nucleosomes with DNA. This is what it represents. This is chromatin, right? Now you also know that at the time of division it becomes rod shaped, short and compact chromosome. So during division, only during division, guys, during division, first of all it will replicate, right, and then it will become rod like short and compact structure, right, chromosome. So what is the hierarchy that it follows during condensation? See, during condensation, so nucleosomes, they form chromatin, but during condensation chromatin will form first chromatin fibers, and chromatin fibers will then become chromosomes. So chromosomes are formed only at the time of division. Otherwise, so they are visible only during division. And what is the best stage to study the structure of chromosome? Let me know. Only at the time of division. Otherwise, when it is not dividing, it appears in the thread-like form. Please tell me these points are clear to all. Please tell me these points are clear to all. Yes, please. Noted? So this is the hierarchy. This is the hierarchy. So as it goes on getting condensed, it forms chromatin fibers and chromosomes. So whenever you use the term chromosome, first of all, it is in the dividing stage, and metaphase is the best stage to study, right? And then understand that chromosomes are formed only when histones are associated. So remember, if we are using the term chromosome, it is eukaryotic in nature. For prokaryotes, we never use the term chromosome. Do you remember the structure of bacteria? What did we say? Prochromosome. The genetic material is called prochromosome, genophore, nucleoid. Remember? We don't call it chromosome. This understanding is going to help us understand few more terms as we proceed in the chapter. Okay? So, yes, you will be able to understand that, decode that. I'll bring that NCERT line in the class. We'll be referring to exact NCERT lines which are so important. Please tell me these points are clear to everyone. And now I would like to proceed to a typical structure of a chromosome. So during condensation, what does a typical chromosome look like? Structure of chromosome. Structure of chromosome. Yes. So see, okay, on condensation. And if suppose it has a sister chromatid also, then it would be like this. Noted? All right. So see. So this part, this part is called the P arm, and the one below that is called the Q arm. Remember? This is the Q arm and this is the P arm. This is after replication. If there would be no replication, then the sister chromatids would not be formed. These are two sister chromatids. Remember? Noted? And we call this particular space as a centromere, the primary constriction. And on either side of centromere, guys, there is a disc-shaped structure present, and this disc-shaped structure is known as a kinetochore. So how many kinetochores around each centromere? 2. Is this clear? So this one is a centromere. And kinetochore, two kinetochores around each centromere. Okay, and what exactly it is? Disc-shaped structures. These are disc-shaped structures for the binding of, for the attachment of spindle fibers. Spindle fibers don't directly attach to the centromere, they'll attach here, attachment of spindle fibers. Of course, that happens during cell division. Is that clear? Noted? And how is the DNA placed within it? So these are These are nucleosomes, tightly bound, right? Tightly bound like this. That's how DNA is present. Okay. So of course it contains DNA. Of course it contains these histone proteins. But when it is about chromosome, then chromosome also contains some non-histone proteins. Understand that. It also contains some, this is a matrix. This is a matrix there. So there are some non-histone proteins also present. Non-histone proteins are also found in the chromosome. NHC we call it, non-histone chromosomal proteins. Please tell me these points are clear to all of you. Yes, please. Yes, guys. Is that clear? Now, we said during division there is condensation. You know, when this genetic material is getting condensed, now condensation can be of two different types. If it gets tightly condensed, then it will have a different manifestation. If it is loosely condensed, then it will have a different manifestation. So during condensation, see, there are two types of chromatins actually. Types of chromatins based on the level of condensation, how much condensed it would be. So we have euchromatin and we have heterochromatin. Euchromatin and heterochromatin. Okay. Euchromatin is loosely condensed. Heterochromatin is tightly condensed. This is the difference. Just a basic difference, but now it will have different results. Okay. So loosely condensed will give rise to, so if it is loosely condensed and if it is tightly condensed, what will it lead to? Now listen, if it is loosely condensed, then this DNA can be transcribed. That means it is transcriptionally active, means it can give rise to mRNA and then mRNA can form proteins. Okay, it is transcriptionally active. This is what NCERT writes. Transcriptionally active. What does this mean? It means mRNA, this DNA can form mRNA. mRNA formed here in front of this DNA, mRNA will be formed. But when it is tightly condensed, it will not open at the time of transcription, so there will be no mRNA formed. So transcriptionally inactive. Transcriptionally inactive. Is that clear? So mRNA will not be formed. And as far as coloration is concerned, when I color, when I stain this chromosome, this one will take, this euchromatin will be lightly stained. It will be lightly stained, while this is so tightly bound that it will take more stain and it will be darkly stained. It would be darkly stained. Please tell me these points are clear to everyone. Yes, guys. Is that clear to everyone? So if mRNA is not formed, it should be not formed. So if mRNA is not formed, it means this DNA is not expressing itself. It is not expressed. How is the DNA expressed? In the form of proteins. So not expressed means it will not form any proteins. It is useless. And if it is forming mRNA, it will form proteins. So it will express itself. So the information coded on this euchromatin will show some phenotype. Please tell me these points are clear. Is this point clear? I am feeling better, Nidhi. I took a lot of rest in the morning, woke up late. So yeah, feeling better now. And I was all ready for this first batch of Phoenix 3.0. Is this point clear? Noted? Should I give you one idea about heterochromatin and the importance of heterochromatin? We know like females have XX condition, right? Females have XX condition, right? Human female is XX. Out of these two X, R1X is euchromatin, so is able to express itself. The other X gets heterochromatized. That's how it happens in a body. Okay, this gets heterochromatized and it becomes inactive. It is inactive. This one is active. And this inactive one is visible as a bar body. This one is visible as a bar body. So basically, in humans, there is one bar body. Males have no bar body. They have just one X and that has to be expressive. Is that clear to all? Please let me know these points are clear to everyone, guys. Yes, please. Tell me this point is clear to everyone. Anyone with any doubt now? Guys, automatically, if I say DNA is acidic in nature, if DNA is acidic in nature, let's see here. DNA is acidic, right? So if it is acidic, it is negative molecule, negatively charged. Histone is basic in nature, so of course they are positively charged. That's about the charge on it. Please tell me these points are clear to all. Anyone with any doubt so far? Yes, guys, anyone with any doubt so far? So next topic. So we have known so much about DNA, but now we have to go in a flashback again. And we have to see how this DNA, how not just DNA, how a genetic material was discovered. So the next topic is discovery of genetic material. So discovery of genetic material. Now this discovery is done by three different scientists. We are going to talk about them one by one. One is Frederick Griffith. Right? Griffith. Then we'll have three other people, Oswald, McAvery, and McCarthy. And then Hershey and Chase. So it will take so many years for them to find these things, and it will be so much fun to go back in the flashback and see the experiments that they made in order to discover these things. Now again, the experiments, the scientists, very important. Then the years. See, Frederick Griffith did this experiment in 1928. Then came these three people, 1933 to 1944. And then Hershey and Chase in 1952. So you have to remember the chronology also. We'll read first about Griffith, then about their contribution, and finally the contribution of Hershey and Chase. But we'll do this tomorrow in our live class on Unacademy Learners app. How all of you can watch that paid batch, the paid live class, I will inform you all in tonight's live session, that is at 12 a.m. So don't forget to join me there, and we'll wind up here and we'll continue with this topic in tomorrow's class. So please let me know, how did you find this class? Let me know in the comment section and also in the chat. And if there is any doubt regarding anything, please feel free to ask in the comment section. And if you have any query regarding the subscription plan, you can again leave a comment section, leave a comment, as well as you can contact us on this 8585858585. Okay, otherwise there shouldn't be any doubt, because all the links are given in the description box, how you can be a part of our successful journey towards NEET. So you can buy this any plan as per your need. And yes, so this is the final batch that that we are starting for NEET 2027. Use this code to get the offer, and the offer is ending today. So that is all, guys. That is all for today's class. I'll see you tomorrow. Where's the live class at 12 a.m.? So the live session at 12 a.m. would be on Unacademy NEET English channel. You have to join me here only. I hope you liked the session, guys. I hope you enjoyed it and you understood the topic. And I'll see you tonight once again with that dhamaka offer and dhamaka opportunity to attend next session also and avail so much more on Unacademy Learners app. So join me tonight, and we'll continue with the chapter on Unacademy Learners app tomorrow 7 p.m. Alright, guys, thank you so much. Tada, bye bye, and see you again at 12.